All right, we're doing Unit 2, Lecture 4, which is titled Metabolism. So this is going to build on the last lecture where we talked about proteins and metabolism, but mostly focused on the proteins themselves. I'm going to start off with a quick review on some of those terms. Metabolism is all the chemical reactions and all the physical workings. Anabolism then are the specific subset that build biosynthesis. These require an input of energy. So when you build things, you need energy. And when you break things down, you actually release energy, which is catabolism. So enzymes and proteins are involved in that. But here we're going to focus a little bit more on the outcome as opposed to the individual structures themselves. So ultimately, metabolism is more complex than what we can break down in a class like this. But from a broad perspective, metabolism is ultimately going to build things up via anabolic pathways and break things down via catabolic pathways. In the end, the breakdown pathways release energy, and the build-up anabolic pathways consume that energy for new purposes. So you're kind of breaking down cellular energy to convert that into ATP, and then spending that ATP to build new structures that fit the body's immediate needs at that point in time. Overall, metabolism can be simplified by this figure here, where you can see some of the different pathways. You have, for example, glucose being catabolized, then precursor molecules can be used for anabolic reactions as building blocks, ultimately being assembled into larger macromolecular structures, which are then the basis and the building blocks for bacterial cells themselves. So remember, some of these macromolecules are similar to your food groups, things like proteins, carbohydrates, fats or lipids, and then even nucleic acids. So ultimately, that model there kind of shows you from a visual standpoint. Going back to enzymes real quickly to review, enzymes have to bind with substrates. These bind with lock and key specificity. Ultimately, an enzyme changes the substrate and then is able to repeat that conversion over and over and over. Eventually, they wear out. Similar to the way a hammer can hit a nail and then be used again and again and again, but eventually the hammer wears out. So if the hammer is the enzyme, the nail is the substrate, there's a reaction there that can be repeated over and over again. So that's kind of what that means. They do not become part of the reaction. A little bit more on the specificity of enzymes. There are some different variations of enzymes. Enzymes are considered either an apo enzyme if it is just made up of protein. Most of them consist of the apo enzyme plus some kind of conjugate, which is either just classified as a cofactor or a coenzyme. So these are then referred to as holoenzymes, meaning that they contain some kind of conjugation. So you have some enzymes that contain organic cofactors, and then those that contain inorganic cofactors, which are very often metal ions and things of that nature. If you've ever looked at a multivitamin nutrition label, you'll see things in there like copper and nickel and heavy metals, very small amounts. But those metals are actually used very often in these conjugated aspects of enzymes, and that's why you need small amounts in your diet in order to make these certain enzymes function properly. Enzymes come in a variety of shapes and sizes. This list here gives you some of the broad classes of enzymes. So hydrolases, enzymes that break molecules with the addition of water. So hydro, water, lase is in reference to breaking or splitting. Ligases, the word ligate means to join together. So a ligase is something that usually joins things, often with the input of ATP. Transferases, the name actually kind of says it. They transfer, but what do they transfer? Parts of a molecule, which can be referred to as functional groups. That's more of an organic chemistry topic in and of itself, but functional groups are parts of a molecule, usually the parts that have specific functions, hence the name. Oxidoreductases are enzymes that transfer electrons. These are part of an oxidation reduction reaction. And then dehydrogenases are enzymes that transfer hydrogens, and they're therefore part of the removal and or transfer of a hydrogen atom, which are key parts of energy processes. So Oxidoreductases and dehydrogenases, these are key enzymes in the synthesis of energy, such as ATP, for example. If you need to pause it here to write those down, we want to go ahead and do that now. I'm going to go ahead and jump to the next slide here. Some examples of those in different classes are listed here in this table. You can go back and look at this table, pause it here. These are just some examples. I put the figure on here so that you can see it. I'm not going to spend time to read all those to you, but these are some examples that you can go back and look at. So take a look at this figure, pause it here if you need to. Okay, naming enzymes, there's a little bit here that's worth knowing. Naming of enzymes is often done where the prefix, the first part of the word, is in reference to the substrate, and then the end part of the name, the suffix, is usually just listed or is described as ace, or is indicated by the suffix ace. This is not 100% consistent, but it's pretty consistent. So what you'll find is that an enzyme often starts off with the substrate and ends with ace. So an enzyme that breaks down lactose, which is a milk sugar, is called lactase. Lactose, lactase is the suffix. Enzymes that break down lipids, lipase, lipases. Lipid, ace, ace. Proteases, enzymes that break down proteins, sometimes more specific in exactly how this, for example, is a class, as it says here. It's not necessarily one enzyme, similar to the lipases. These are more broader classes, but these are enzymes that break down different types of lipids and different types of proteins. So anyway, that's just a real quick note about those. So if you go back and look at some of these examples here, penicillinase, for example, breaks down penicillin, right? DNA polymerase is one that joins together parts of a nucleotide. So that's not something that you have to commit to memory all the different examples, but if you understand ACE and that the substrate is usually named in the first part of it, you can actually kind of guess, even if you don't know what an enzyme does. You can kind of guess what it does just by knowing that alone. Okay, a couple more things about enzymes, and then we'll get into the actual process of getting energy from the cell. Enzymes are either produced on a regular basis or on an on-off sort of need-based situation. So constitutive enzymes are those that are effectively produced all the time. There are many different types of enzymes in the body that you will need fairly routinely. These would be enzymes involved in various detoxification processes involving oxygen. We talked about in the last chapter that the body is constantly dealing with oxygen byproducts, superoxide dismutase and singlet oxygen, for example. And so you have different enzymes that are involved in oxygen processing that are always required as long as you're breathing. And so these would be examples of constitutive. You have other enzymes that are regulated and turned on or off depending on the need of the cell. These are often in relation to things like your food substrates. Food is not something that you eat consistently all the time, and so depending on the kind of food substance that you're looking at, you may only produce that enzyme if the food is available in your body. In other words, the enzyme doesn't get turned on until the food is in the body. So those are examples of regulated enzymes. Okay, next what we want to talk about, stepping outside of enzymes and proteins, is just how the cell is extracting energy from the food that we eat. What we want to understand is that at every step along the way, there's some kind of protein and or enzyme involved. For this class, we're not going to sit and name and have you memorize every single one. So what I wanted to do is show you up front that enzymes and proteins are involved, give you some examples, show you how generally they work. For a class like this, we're not going to try to memorize every step and have you memorize every enzyme and exactly what it does. So what you want to understand is that there's always an enzyme involved in some way and at every step. Okay, and so from here we can kind of then extrapolate that these processes are conducted by enzymes in a series of steps, and then we're kind of taking a step back from the details at the micro level and looking from a little bit broader perspective what's going on here without emphasizing too much of every little name and enzyme involved. So ultimately energy comes into our body through our food, and everybody knows that at a certain level. The question is, what is it about the food that gives you energy? And the answer ultimately comes down to the fact that energy lies within the chemical bonds of our food. there's always more to it than that. But ultimately, if you look at, for example, a carbon and a hydrogen atom, there is a physical energetic force that occurs between these two things. And it literally takes energy to form them, and then energy can actually be released when these two are broken. So energy input to form, energy release upon breaking. So going back to the idea of catabolic and anabolic, this here represents a catabolic reaction to release energy, and this here represents an anabolic reaction to form energy. Okay? This is often done through carbon-hydrogen bonds and or carbon-oxygen bonds. One of the things about carbohydrates, which are entirely made up of carbon, hydrogen, and oxygen, is carbohydrates make a really good energy source for our bodies. One of the reasons why our bodies like carbs, at least in terms of quick energy, is because they're easy to break and they're easy to form to a certain extent. So these can be done with other types of bonds, but those are the ones that our bodies prefer. So ultimately, when you eat food, what happens first actually is that you'll break the food down to release energy catabolically. Then what will happen is your body will extract that energy and tie it up in intermediate molecules called ATP. ATP then is almost like cash. It's very often referred to as cellular currency. It's kind of like the way you could sell your textbook and then cash it in for physical cash or digital cash, and then hold on to that and then later go spend it on something else. ATP is kind of like that, where the body can use food to extract energy, tie up that energy in a molecule, and then go spend that molecule wherever it's needed later. So energy is used in this process by breaking and releasing these bonds. Sorry, this slide's a little out of order there. But it's important to note then that there's a difference between metabolism and digestion. It's easy to get those terms mixed up. You may actually think there isn't a difference. But digestion is actually the process by which the bigger macromolecules in your food, these are like the proteins and the polysaccharides, which would be like starches and larger sugar molecules. These are first broken down in stages through digestion. In other words, macromolecules or polymers are broken down into monomers, individual units. Digestion is something that takes place outside of your cells. This is something that depends on the kind of organism. Remember, we talked about extracellular digestion in the previous chapter, where bacteria and fungi release their enzymes outside of the cells. That basically is what you're seeing here. Macromolecules are broken from chains down into monomer units. Then, once the food is brought into the cell in smaller pieces, then metabolism takes those monomers and breaks them down step by step and releases the energy, as it shows in this figure, from the chemical bonds in each monomer. So macromolecules or polymers consist of many monomers. Each monomer consists of many individual carbon-hydrogen, carbon-oxygen bonds, amongst others. And those bonds then, as we said in the last slide, contain the energy. So digestion is really about getting the macromolecules into a monomer form so that they can go into our cells. Mechanical digestion is basically you chewing your food. Chemical digestion is you using enzymes to snap these bonds and break them down. Sometimes this happens, I should say, it typically happens at the same time. For example, in the mouth, you may have heard that digestion occurs in the mouth. That's quite literally true. So chewing your food is a mechanical, but also chemical digestion does begin in the mouth with an enzyme like amylase, for example. Amylase is an enzyme that breaks down carbohydrates into sugars, and also proteases break down proteins into amino acids, as we mentioned. Those can all occur in the mouth. Some of those then occur more in the stomach, such as proteases. So from the mouth to the stomach, you're mostly focused on things like digestion. And then once the food, the monomers, get into the small intestine, they're absorbed into the small intestine cells, which they're metabolized inside the cells and then passed throughout the body to be metabolized in other types of cells. Ultimately then, that digestion allows us to make small little packets of energy that can be transported and then ultimately sort of synced up through the body where they're needed. And so this often occurs through the blood, where the individual molecules pass from the bloodstream into our cells, through the cell receptors, into the internal intracellular part of the cell, where they're then metabolized step by step by step to make energy. So a little bit more about this energy being extracted. As we said, the food contains chemical bonds, and the chemical bonds contain energy, which is, in this context, you could refer to it as potential energy. Potential energy is like energy at rest. What we end up doing with our body is, as we break those bonds, we actually extract some of the electrons and even the protons, the subatomic building blocks of the actual molecules. And although we're skipping a lot of the details here for a class like this, what ends up happening is those protons and electrons end up being used as their own potential energy to build up and charge a molecule called adenosine triphosphate, which is ATP. ATP stands for adenosine triphosphate. This is the equivalent of cellular currency or cellular money. It's a storehold of value, or in this case, a store of energy. So the food contains potential energy. That potential energy is then converted into ATP. Your body then stores ATP, kind of like you hold on to cash, and then it can spend it wherever it's needed. ATP is kind of like cash. You can use it in a variety of places. Not necessarily everything, but a lot of things can use it. So ultimately, you can do things like active transport. If you go back to the previous chapter, there's a difference between passive transport versus active transport. Active transport requires energy. That comes from ATP. Anabolic reactions, if you go back and look at the very beginning, it said that they require an input of energy. That energy is ATP. By the way, there's always little exceptions, but generally speaking, it comes from ATP. Muscle contraction. For humans and animals, mammals specifically, we spend a huge amount of our energy just by muscle contractions. So moving muscles, plus many, many more. So those don't even begin to cover all the ways. But ultimately, your body consumes huge amounts of it. So ATP here is ultimately a molecule called adenosine triphosphate. Adenosine is essentially the backbone that you see over here. And then the term tri stands for 1, 2, 3 phosphates. So adenosine triphosphate. It's a strange little molecule, but ultimately it can be thought of like a rechargeable battery. And the adenosine, the base, is kind of like the battery casing in and of itself. And the phosphates are kind of like the juice that go inside of it. And kind of like a rechargeable battery, you hang on to the casing, and then you ultimately just fill it up and deplete it as required. And that's kind of how ATP works. So ultimately, you can use ATP by breaking off one of these phosphate bonds. Usually it's the first phosphate bond, sometimes it's the second phosphate bond. In rare cases, you could actually even break off the third one, but usually it's only the first and or second. So in other words, you don't usually run the battery all the way down to zero. You usually charge it before it gets completely dry. But ultimately, you could theoretically do that. But the phosphates are broken off, and as you break a phosphate, it releases energy. And it then requires energy from your food to put a phosphate back on it. So kind of like you got to stick the rechargeable battery into the wall and pull some juice out of the electrical outlet to charge it. Instead of having electricity, this is where the body's food is what required to replace these phosphates, like filling up the empty battery recharging. So every single day, every single second of our life, ATP is being charged up and broken down with different molecules all the time. Depending on who you ask, you'll get different numbers on this, but it's estimated that we use roughly 100 to 150 pounds of ATP each day. And considering that these molecules are so small, you can't even see them with the most advanced microscopes, that's pretty amazing. So essentially you're using almost unquantifiable amounts of ATP every day. Numbers that are difficult to even put in perspective. So pounds, hundreds of pounds, over 100 pounds of ATP every single day. So the amount of it is really pretty stunning. So every day you're breaking these bonds and then recharging them, kind of like using your battery and then refilling it back up. So just to reiterate the difference here, digestion sets up your cells to create the units to then feed ATP. Metabolism is the process by which the ATP is created. So digestion actually begins with the cells breaking food down, ultimately to feed those little particles into your cells so that the particles themselves can make ATP. What's interesting about digestion is that you're not actually getting any energy from digestion. You're setting yourself up to get energy later. You might think of digestion as like working 8 hours at your job, but you haven't gotten a paycheck yet. You should get a paycheck and you will get a paycheck, but you haven't gotten it yet. And that's kind of like digestion. And then metabolism is the process by which you actually put the money in your bank account. So ultimately, digestion can actually, in some cases, require energy, kind of like maybe you have to spend gas to get to work. You actually maybe require a little bit of input in order to then later get more out of it. Hopefully you're going to make more money at your job than what you spend on gas, but you do have to put some energy into it. And by the way, this is one of the reasons, although there's several, one reason why you can sometimes feel tired if you eat too much food is because you're actually requiring an input of energy to digest it. And so you actually haven't got any energy out of it yet, and you're actually inputting energy into the food to help break it down. So metabolism is ultimately where you get that energy later, but that takes time, and digestion has to occur first. In this figure here, I put that in your handout there just to look back at it, just as a quick visual that you can go back and look at there later. Okay, the next part here is how do the cells make ATP? And for this class, we're going to keep this real simple. This is a whole lecture that you can get into many, many weeks trying to figure out each step of these pathways. What you're looking at here are 3 catabolic pathways, and they're referred to as aerobic respiration, anaerobic respiration, and fermentation. Each one of these pathways consists of many, many steps, at which each step a different enzyme is used to break down different food particles at different stages of metabolism. So very often these start with glucose, and then glucose is broken down step by step by step until in the very end a certain amount of ATP is made, and ultimately the glucose is converted into ATP. Aerobic respiration occurs with oxygen. Anaerobic respiration is nearly the same pathway, except it occurs without oxygen. So rather than breathing in O2, you use some other kind of molecule. Fermentation is different from aerobic and or anaerobic in that it doesn't require oxygen, but it also doesn't use all of the steps in the pathway. So respiration pathways end up breaking down the food completely so that there's essentially nothing left. Fermentation is an incomplete breakdown, so that as you break it down, you extract some energy, not as much, and as a result, you end up leaving byproducts at the end that are like used as a way to kind of clean up and recycle some of the energy inputs to keep the process going. This is one of the ways that we create alcohol, for example. Alcohol is actually a byproduct of fermentation. The cell that's making the alcohol doesn't want it. It's something that they're actually getting rid of. But the alcohol is something that we can then consume. But fermentation is something that leads to an incomplete breakdown. When you have aerobic respiration, you will never get alcohol. This, by the way, one of the reasons when you create alcohol, you actually have to seal off the jar, and you have to make sure it's done anaerobically. If you try to make alcohol aerobically, you'll never get any. So when you have fermentation, there's no oxygen, but there's also an incomplete breakdown, and that's where anaerobic respiration is a little bit different. Anaerobic respiration, also without oxygen, breaks it down completely, gets more energy, whereas fermentation breaks it down incompletely. You have leftover byproducts at the end, and not as much energy in the process. So those are ultimately some of the ways. So a little bit more about aerobic and anaerobic. Ultimately, when it comes to aerobic respiration, molecules like glucoses and proteins and lipids are completely oxidized, meaning that all of the energy is extracted out of them. And at the end of the process, oxygen in and of itself is used to kind of clean up the process. It's described as the final electron acceptor. So the electrons are the little subatomic particles that are passed off from your food. These are part of the chemical bonds that hold the food molecules together. So the electrons are part of those chemical bonds that hold things together. And as they're passed around, they have to go somewhere before the final process is over. And oxygen is ultimately where the body dumps those electrons. And if you stop breathing and you stop having oxygen, you'll be unable to continue to metabolize. Your body will start to build up with harmful byproducts, and you'll have cellular death very rapidly. Ultimately, the way that you and I and heterotrophic organisms function is through aerobic respiration. Anaerobic respiration is much of an exception to the rule. Ultimately, some organisms have evolved to pass off electrons to something besides oxygen, and these are much more of an exception to the rule. But when it comes to microorganisms, as it says down here, this can happen, and ultimately, rather than breathing oxygen, they can pass these electrons to things like nitrogen and sulfur compounds. And so that can allow them to extract more energy than they could through fermentation, but not require oxygen at the same time. So as I mentioned, when it comes to respiration, either aerobic or anaerobic, food molecules are completely oxidized. That is to say that all of the energy has been extracted out of them, and the only real byproducts that are left behind are carbon dioxide and water, CO2 and H2O. And interestingly, as you metabolize your food, that is really where the majority of the energy actually, the majority of the byproducts go, is you actually breathe it out as CO2 from your lungs, and or it becomes part of what's called metabolic water inside of your cells. It makes up roughly 10% of the water in your cells is metabolic water. So you breathe out most of that carbon, and your cells get partially filled up with the water itself. When it comes to fermentation, on the other hand, you'll have a lot of material still left inside the cell. So it's considered incomplete oxidation. So rather than it being broken down into carbon dioxide and water, you'll have byproducts depending on what type of fermentation, such as things like ethanol, which is an alcohol, or lactic acid, and a whole variety of different acidic compounds. There's a lot of different fermentation processes, but they always end up with an incomplete oxidation and a byproduct of some kind being left behind. Ultimately, fermentation does produce energy, just not very much energy. It's a small fraction compared to what you would normally find from respiration. So ultimately, your body can make energy through a couple different ways. For you and I as humans, it's aerobic respiration primarily, and a little bit of fermentation to fill in some of those gaps. This, by the way, is what happens when you get when you consume lots of energy during exercise, especially a lot of aerobic exercise, but also anaerobic exercise, weightlifting too. You end up creating lactic acid. You may be familiar with lactic acid is a byproduct of exercising, usually muscle fatigue and things like that. But lactic acid is one of the byproducts that your cells produce through fermentation if you're not getting quite enough oxygen. So usually it's when you're breathing heavy, you've been working out hard, and you don't have quite enough oxygen, your body will convert into fermentation in your cells, and lactic acid will be produced as a byproduct. Now, when it comes to microorganisms, fermentation is something that we end up using in a variety of ways to create various types of products that are useful. So the waste, as it says here, from one organism can actually be used as sort of like a treasure for another, if you will. But various types of food products here mentioned require fermentation to create them. And these generally require a lack of oxygen or sealing it off in some way or closing the organisms in the process so that they don't have access to oxygen. So alcohol, cheese, certain types of breads like sourdough bread, for example, and a variety of fermented foods. There's a bunch of different types of fermented foods out there. Pickles are technically a fermented food. Kimchi, fermented cabbage. You can actually ferment nearly any kind of vegetable. There's a process to it, but usually you just put the vegetable in water, add salt to a certain ratio, and then seal it off. You just close off the jar, and it will eventually ferment, and it will kind of create a different texture and taste. And very often they're healthier for you, assuming that you do it correctly. So as I mentioned, processes like aerobic respiration are really a series of steps. They're metabolic pathways. And even though we're not going to name every step, there are many steps, many different reactants, many different enzymes. So a pathway is just really the same concept of an enzyme and a substrate, but what ends up happening is as the first enzyme reacts with the substrate and changes it, it sets it up to then be acted upon again by another enzyme. So the product of the first enzyme becomes the substrate for the next enzyme, kind of like an assembly line. And so each little worker in the assembly line chain modifies it and then passes it to the next. And that's essentially the aspect of a metabolic pathway. There's a different kind of enzyme at each step, and the enzyme can only function once the previous step has been completed. So as it says, the product of one reaction is often the reactant substrate for the next. Now, metabolic pathways get complex, and as I mentioned earlier, this class is not really meant to go into a super great amount of detail about all the nuances of metabolic pathways. So we're just trying to give you more of a bird's eye view without all the detail here. So a little bit goes a long way, in my opinion. But pathways can have a variety of different layouts. You have linear, and then you have branching and cyclic, meaning they cycle back to the starting point. So branching pathways are pretty much the norm. They start off in one direction and then can alternate in different directions depending on the needs of the cell. Some of these, one in particular called the Krebs cycle, is cyclic. Cyclic pathways end up converting the very last step of the cycle back into the first step, and it ends up then starting over almost in a circular kind of fashion. And each one of these steps does not exist in isolation. All these paths can interconnect. So I'll show you what that looks like here. So linear pathways would simply go from one to the next until you reach the end point. Those happen, but more often you have divergent and or convergent, where one product can be converted into a multiple different number of endpoints. That depends upon the needs of the cell. And why these are more common is because the cells are adaptable. Depending on what you're eating, depending on how much you're eating, you can convert products down different pathways depending on what makes sense right now in the cell. Cyclic is just a different way of describing a pathway that doesn't end with the given output. It ends by cycling back to the beginning. So if it were linear, rather than ending with E, it would cycle back and it would start over again with A. So cyclic are a little more confusing, but basically the idea is that it ends up coming back to the beginning so it can happen over and over and over again. So these are the ways that pathways kind of work when one goes to the next. You can go straight. Divergent, meaning that it branches from one point into multiple endpoints. Convergent is the opposite, where starting from two different beginning points, you can end at the same point by starting from two different locations. So at every step along the way, cells in your body are using different pathways for different purposes. Aerobic respiration, to go into a little bit of detail, ultimately employs three major types of pathways that are all part of aerobic respiration. So glycolysis is somewhat of a linear pathway, somewhat of a divergent pathway. Glycolysis ultimately starts with glucose. This is actually the cell's preferred energy source. It's one that the cell can extract energy from the easiest. Certainly not the only one, but if you have sugar in your cells, it will convert the sugars into energy first. So glycolysis is the pathway by which the cells will use the glucose energy. The products of glycolysis can be used exclusively, or the byproducts can be shuttled into the Krebs cycle. Krebs cycle is one that allows us to really create the precursors for ATP. I didn't get into it in this lecture, but they create what are called electron carriers. Electron carriers, as the name implies, carry the electrons, which are some of the particles from our food, ultimately to the last step of the process, which is called the respiratory chain. These electrons are then shuttled through a series of carriers to ultimately to make ATP, which occurs inside of our mitochondria. And this is why the mitochondria is referred to as the powerhouse of the cell, because it's able to use the products of food to make ATP. This figure here shows a mitochondria diagram, and it shows these pathways in reference to where they're occurring. Now, we didn't talk about pyruvate oxidation. That's sometimes called an intermediate step, but this shows here glycolysis. The citric acid cycle, I apologize, is also referred to as the Krebs cycle. I mean, for that to be confusing. Citric acid and or the Krebs cycle, same thing. This is an example of a cyclic pathway. And then ultimately, the electron transport chain is where most of your ATP is created. So this is not getting into all the nitty-gritty details, but showing that as food comes in, outside of the cell, it's pumped into the cell for the Krebs cycle and the electron transport chain. And at every step of the way, a little bit of energy is created, but most of the energy is created at the very end via the electron transport chain. Now, as it says, go back here just real quick, glucose is the preferred energy of the cell. So if you were to eat lots of carbohydrates, lots of sugars and things like that, your body is going to break those down first. However, we don't just eat carbohydrates, and ideally we shouldn't be eating too many carbohydrates, certainly not simple sugars. So ideally you have a fair amount of protein in your body, and God forbid fat. Fat has been largely demonized, but good fat is actually relatively healthy for you. So if you're eating proteins and fats, your body's going to have to break those down too. So rather than starting at the very beginning of the process, what ends up happening is fats and lipids are broken down into intermediates and then shuttled into different intermediate points along that pathway. And that, kind of going back here real quickly, is where different convergent pathways can end up in the same place. So you can actually start with glucose potentially, or a protein, or a fat, and all end up at the same endpoint, which in this case is ATP. And that's kind of the idea behind alternatives to glucose. Now, rather than lecturing on this, I put together a handout for you, so make sure to look at the alternatives to glucose handout, and there's some questions that go along with that in the study guide. So make sure to look at that. It's relatively short, but it gives you a little bit more detail about kind of how that works. And this figure is on that handout, which diagrams some of the ways that different compounds are converted into these pathways via different intermediate points. I'm not going to ask you anything real specific about all the details of each step, just sort of general questions like you see on your study guide about how these interact in different parts of the cycle. Last couple of things here about the cycle of the path is that our bodies have essentially the ability to control these pathways kind of like a gas pedal or in a brake almost. We can actually control pathways so that they don't just run endlessly. Ultimately, the cell doesn't have a brain, and therefore it has to have a different way of controlling things. You and I have more complex feedback. If we eat too much and we get sick, we stop. But at the cellular level, that's not really how it works. And a cell needs to have a different way to be programmed so that it can turn on or turn off different elements depending on what makes sense at that point in time. So for example, if you've just eaten 1,000 calories of a sugary meal, then you're going to need to make sure you have enzymes to break that down. If you haven't had any food for the last 18 hours, now you got a different problem. Now you're going to need to turn on enzymes to maybe start breaking down some of your fat. And now you're going to need a different mechanism than you do if you have 1,000 calories of carbohydrates. So at any point in your life, at any point on a per cell basis, there are different factors that are happening. And so these are sort of examples about how the cell can kind of dictate what's happening. So competitive and non-competitive inhibition, these are mechanisms by which the substrate and the enzyme can be affected once the enzymes are produced in the cell. Genetic regulation is a process that actually determines when and where, if the enzyme is produced in the first place. So we'll take a look at competitive inhibition and non-competitive inhibition. These terms are related to the way that the substrates can interact with the active site. So competitive is in reference to the active site. So competitive inhibition is a scenario where the cell has a competitor that is similarly shaped to the normal substrate, and they're both able to bind to the active site. The difference is that the competitor is not reacted upon. It's sort of like sticking the wrong key in a keyhole, but it might fit into the keyhole but doesn't open the lock. And so it just blocks the regular key from fitting in there. That's kind of like a competitive inhibitor. So these are ways that the body can slow down an enzyme. If that competitor is blocking the active site, then the normal substrate can't bind, and as a result, the reaction with the normal substrate doesn't proceed. So as you can see here, normal substrate binds, reaction creates products. If we have too many products and we don't need that, then oftentimes a competitor will block the active site until those products are used. And it's really just a temporary mechanism. It allows the cell to not produce too many products when they're already in excess. Non-competitive inhibition is a little different. Non-competitive inhibition is a scenario where instead of a competitor competing at the active site, the cell will produce a different molecule called a regulatory molecule. This is also referred to as an allosteric molecule. And ultimately, it changes the active site through binding at a different site called the regulatory or allosteric site. The end result is the same. The product and the reaction are temporarily turned off or halted, but one involves competition through the active site, and the other involves a different mechanism, not via the active site. So as I mentioned, competition refers to the active site, so competitive and non-competitive refer to how the enzyme is being acted upon in relation to the active site. So in relation to competitive and non-competitive inhibition, what's really interesting is that very often the molecules, the competitor or the non-competitor molecules, are actually molecules that occur somewhere in the metabolic pathway. And what generally happens is that one of the end products of the pathway ends up acting as either a competitor or a non-competitor, which is a regulatory molecule, by binding or interfering with the active site. So some of these competitor molecules are just in products. They're products that occur near the end of the pathway. And what happens if they become too numerous, they start to build up. And once they build up to a high enough concentration, they'll literally start to interfere with some of the enzymes early in the pathway, either by blocking the active site or acting upon a different part of the enzyme called the regulatory site, or what's also referred to as the allosteric site. So these get technical in terms of how exactly they work, but ultimately what ends up regulating the molecule, the enzyme, is one of the products of the pathway. And it's fairly ingenious, because what ends up happening then is as the end product becomes numerous, that implies that you're not using it. It's almost like you have way too many jars of peanut butter in your pantry. So it doesn't make sense to keep buying it, right? The product is too numerous. We don't need any more of it, right? So your cells now have a mechanism for that product itself to effectively turn off the very pathway that makes that product. And this is where the exact mechanism is either competitive or non-competitive. Ultimately, it achieves the same goal. The cell has a couple different ways of doing that, but these are examples of negative feedback. The feedback of the end product negatively down-regulates the very process that creates it. Now, this occurs only for a short period of time. Kind of like you take peanut butter off of your grocery list, right? And then a year goes by and now you don't have any more, so you put it back on your grocery list. So it's a temporary process that only occurs until the end product is once again needed, and ultimately the inhibition is released and the process continues. So it's very rarely a permanent process. And as it says here, the goal here is a reduction of waste. Kind of like you don't spend money on something that you have lots of in your pantry. And the last thing here is that this can also happen at the genetic level. So competitive and non-competitive inhibition occur when a molecule has already been produced. The protein or the enzyme already exists inside the cell. But when it comes to genetic regulation, you can have enzyme repression or enzyme induction. And as the name implies, this actually occurs in terms of repression, for example, at the genetic level, such that the enzyme itself is never produced until it's needed, or turned off once it's no longer needed. So repression is essentially a more fundamental process. Rather than blocking the enzyme itself, you go all the way back to the DNA level where the enzyme is actually produced, and you turn it off at the genetic level. And so ultimately, this is not as quick as the previous processes. It's more of a long-term process, but it gives you more of a complete control by turning on or off the very process that makes that enzyme in the first place. So repression turns it off, enzyme induction turns it on. There are presumably many examples of this in the body. these types of processes are difficult to study. These have been done in bacteria, for example. So the tryptophan regulation pathway is one that has been studied fairly well. Tryptophan is an amino acid, and tryptophan itself can actually turn off some of the enzymes that degrade tryptophan or catabolize tryptophan. So when the bacterial cells that have been studied, they've been shown that when you give them lots and lots of tryptophan and there's an excess in the cell, tryptophan itself will bind to the DNA and turn off the enzymes that can actually synthesize or catabolize various tryptophan products. And so it's essentially a fundamental way of turning on or off what's needed in the cell all the way at the genetic level. All right, that gives you some ideas about metabolism without going into every little step of the pathways. So make sure that you finish, if you haven't already, watching the videos I've given you. You've got a couple different videos this week. And then make sure you take a look at the alternatives to glucose handout and go back and review the questions on the study guide.